Greetings to everyone. Welcome back once again to this educational channel on biology. In this video, we are going to discuss techniques of answering biology paper 3 KSSM syllabus, and we are going to continue our discussion on experiment 1 on osmosis, which is based on experiment 3.5 from your form 4 textbook, page 63, right? So in this experiment, we soaked the potato strips in sucrose solutions of different concentrations, and then we determine the percentage change in mass of the potato strips, right? So in order to find out which solutions are hypotonic, hypertonic, or even isotonic, okay? So this is a continuation, this video is a continuation of the previous video on experiment 1 on the potato strips, but this is part 2, where we will discuss other science process skills like stating the hypothesis, stating the operational definition. So I have invented a code or a memory aid to help you easily give the operational definition during the exam, to remember what the components are, right? So stay tuned. And also we'll discuss other types of questions related to the science process skills that can be asked in biology paper 3, which is the practical test, right? In order for you to score for your biology paper 3. So let's get started. Let us recap and review what we have discussed in the previous video concerning the experiment involving osmosis. Our aim was to study the effects of different concentrations of sucrose solutions on the mass of the potato strip, and the procedure was as follows. First, we get a potato, and then using a cork borer, which was pushed into the potato, we produce 3 potato strips, which are cylindrical. After that, we place each potato strip on a white towel, and using a knife, we cut it so that it's 5 cm in length. Using a ruler to measure the potato strip, so that is 5 cm in length. Next, we place each potato strip on a filter paper and dry it. After that, we place the potato strip on the electronic weighing scale or electronic balance and weigh it to obtain the initial mass of each potato strip. Next, each potato strip is put into a sucrose solution. So A, B, and C are sucrose solutions of different concentrations. So we soak the potato strips, okay, 1 in each beaker. Soak the potato strips in the sucrose solutions A, B, and C of different concentrations. Okay, not soak them in water. Okay, I'd like to clarify this from the related to the previous video. Okay, soak the potato strips in the sucrose solutions A, B, and C, not in water. And then we start the stopwatch. Okay, and after 20 minutes, we take out each potato strip, dry with a filter paper, and weigh it again to obtain the final mass of the potato strip. Okay, so the important thing here is don't confuse the 3 potato strips. Don't confuse the initial and final mass of the 3 potato strips. For potato strip in beaker A, you should record it in the corresponding column for sucrose solution A. And don't mix up with sucrose solution B and C. Okay? So what I mean is that do the experiment systematically and record each value of the mass of potato strip one at a time. Okay? After you have weighed them. Now, before we look at the results again, let us revise our concept, the concept of osmosis, right, from chapter 3, form 4 textbook. So, if we have 2 solutions of different concentrations, such as A and B, water will have to diffuse from one region to the other. So, where is the direction of diffusion? Right, if you look at A is, compared to B, A is a hypotonic solution. Hypo, the po here means low. Hypo means under, so it's low. Low what? Low solute concentration. For example, these two red molecules are the solutes, right? Solute molecules. It could be sucrose, it could be other substances like salt and all that. So there are only two molecules here, two molecules of the sucrose, whereas on this side, B, there are many more, right? So we say that relative to B, A is a hypotonic solution, meaning it has low solute concentration, but high concentration of free water molecules, the blue one, right? Okay, when it has low solute concentration, it will have a high concentration of free water molecules, the free water molecules that can move around. And we say that it has high water potential. Okay, so loosely, we can refer to this high water potential. You can have this idea that it has high concentration of water, the free water molecules. So because textbook use the word high and low water potential, so we have to know this word. So a hypotonic solution has low solute concentration but high water potential. And in the common way of calling it is dilute solution. It is a dilute solution relative to B. Okay, so B on the other hand is a hypertonic solution. Look at this word E, hyper. E stands, we can imagine it stands for elevated. So it has elevated or high solute concentration. It has high solute concentration. So the hyper means over or high solute concentration, such as high sucrose concentration. But when it has high solute concentration, it will have low concentration of water. All right. Now, why is that so? Because some of these water molecules, which are polar molecules, they'll be attracted to the sucrose molecules and they will cluster around the sucrose molecules. Not seen here, okay? But imagine that the blue water molecules cluster around the sucrose molecules and they are no longer free, So they cannot move around, and this will effectively lower the concentration of water, free water molecules in B, when the solute concentration is high. So when it has low concentration of the free water molecules, we say that it has low water potential. The potential for it to diffuse to somewhere else, to move. To move is less. Okay. Now, then it is considered a concentrated solution. Okay. So basically, we have to know that what the meaning of hypotonic solution is. That means a solution of low solute concentration and high water potential. Okay. Following what is said in the textbook. And for hypertonic solution, it is a solution with high solute concentration but low water potential. All right. So the osmosis rule is that all water will always diffuse from an area of high water potential or high concentration of water. Right. This we call a hypotonic solution, which is a solution of lower solute concentration or a dilute solution, to an area of low water potential, from high water potential or high water concentration to an area of low water potential or low water concentration. And that is the hypertonic solution that is concentrated. So it has higher solute concentration than the first solution, and it is also called a concentrated solution. Okay, or we can say water diffuses from hypotonic solution, always diffuses from a hypotonic solution like A into a hypertonic solution like B. Okay, from dilute to a more concentrated solution. So for our potato experiment, the cell sap of the potato may be hypotonic or hypertonic to the surrounding fluid. Okay, so let's look at the results again. Let us refer back to the results that we obtained in the experiment 1 in the previous video, the experiment on osmosis. So we have the sucrose solutions of different concentrations, A, B, and C, right? But the concentrations in this case are unknown, not known, right, for this experiment. Then here's the initial mass and then the final mass of the potato strip after soaking it in the sucrose solution. And then from there we calculated the percentage change in mass according to this formula. So what can we conclude from the results of our experiment? So here, from the values for the percentage change in mass, we can conclude whether the solution is hypertonic, hypotonic, or isotonic, right? For example, for A and C, we can see that the percentage change in mass is a negative value. negative 6.5 and negative 14.3 respectively. So let's explain how we get a negative value here, right? So when we see the negative value, it means that for the percentage change in mass, it means that the potato strip has lost water and lost some mass, right? So it means that the sucrose solution in which the potato strip was soaked is a hypertonic solution. Hypertonic solution, that means a more concentrated solution, having a higher solute concentration than the cell set of the potato cells. In this case, the solute is sucrose, sucrose for this solution. So when the potato strip is soaked in A, okay, we say that A is hypertonic to the cell set of the potato cells, all right? So this type of concentrated solution will draw water out from the cells by osmosis. So water diffuses out of the cells by osmosis into the hypertonic solution, and thus the potato strip loses water and loses some mass, right? So the final mass of the potato strip will be less than the initial mass, and thus when we calculate the percentage change in mass, final mass minus initial mass here will be a negative value, right? So the percentage change in mass will be a negative value, right? So if the percentage change in mass is a negative value, like for A and C here, we conclude that the solutions are hypertonic to the cell set. That means having a higher solute concentration than the cell set of the potato cells. Or in other words, having a lower water potential or lower water concentration than the cell set. Okay, it's a concentrated solution. Whereas for B, we can see that B has a positive value for the percentage change in mass, 10.1. So we can conclude that water has diffused into the cells by osmosis, and that the potato strip gains water and gains mass. Thus, the final mass of the potato strip is more than the initial mass, right? Okay, so we can explain this way. The sucrose solution B is hypotonic to the cell set of the potato cells, meaning it has a lower solute concentration than the potato cells. So according to the osmosis rule, water diffuses from the hypotonic solution to the hypertonic solution by osmosis. Thus, water diffuses into the cells by osmosis, and the potato strip gains mass, right? And gains water and gains mass. Right. Then how about isotonic solution? What happens in an isotonic solution, right? So an isotonic solution is a solution that has the same solute concentration as the cell set of the potato cells, okay, of the potato cells. Thus, water diffuses into the cells and out of the cells at the same rate, So there is no net gain or net loss of water from the cells. Thus, if the initial mass of the potato strip is 2.30, the final mass should be the same too, right? And the percentage change in mass is 0. Okay? So I hope you know how to explain, you remember and know how to explain what happens to the potato strip in the different types of solutions. Right, now in our experiment that we discussed just now, we did not know the concentration of the sucrose solutions, right? But for an alternative question, let's say that you were also told the concentration of the sucrose solutions. Then you can plot a graph of the percentage change in mass of potato strip against the concentration of sucrose solutions. Okay, for example, let's say that you were given 3 sucrose solutions, 0.2 m, 0.3 m, 0.4 m sucrose solutions. So you can find the percentage change in mass. For example, here for 0.2 m, it is around 23%. Percentage change in mass is 23%. For 0.3 m, it is about 9%. And for 0.4 m is a negative value around, say, a negative 11 or 12%. Negative 12%. Okay, then you can draw a line through these 3 points, and the curve will cut the horizontal axis here. Okay, that is significant. All right. So here I show you a graph which has more points. And then after plotting the graph, you can see that if it's a curve that cuts through the horizontal axis. Okay, so you need to understand the concept behind this graph, all right, and explain the graph. So how to explain the graph? From 0 to 0.32 or a bit here, right, until 0.32 or more, a little bit more than that, the solutions are hypotonic solutions. Why do we say that? Because there is a positive change in mass, okay, meaning to say that water diffuse into the potato cells by osmosis from the hypotonic solutions, all right, down the concentration gradient, and this caused the potato strip to gain mass. That's why there is a positive change, or there is an increase in mass here. Okay, and then at the point where the horizontal axis and the curve meet here, the intersection here, right? There's no change in mass here, right? For this point. So this is the isotonic solution, where water diffuses into and out of the cells at equal rates, and there is no change in mass. Okay, so we must know, we must be able to determine this value. For example, this could be at 0.33 m, the intersection here. So we can say that in 0.33 mole per decimeter cube or 0.33 m sucrose solution, there's no change in the mass of the potato strip. Thus, at this concentration, 0.33 m, the sucrose solution is isotonic to the potato cell. Okay, when there's no change in the mass of the potato strip after soaking it in the isotonic solution. Okay, but beyond 0.33, right, we can see that the percentage change in mass are negative values. So this means that there is a decrease in mass after soaking. There's a decrease in the mass of the potato strip. So all these solutions beyond the point of intersection, they are hypertonic solutions, more concentrated than the cell of the potato cells. So water diffuses out by osmosis, and then the potato strip lost mass. Okay, and the percentage change in mass is negative, meaning that the final mass of the potato strip is less than the initial mass. Okay, because some water has been lost from the cells. Right, so I hope you understand this curve, and just visualize how it looks like. The hypotonic solutions are here until the intersection, and then at the intersection, this is the isotonic solution, and after that, beyond that, all these are hypertonic solutions. Okay, so this kind of graph can be asked also in the objective questions, and maybe even the structured or essay questions. So take note now of the concept behind it. Let us go on to other possible questions related to this experiment on osmosis, which is also related to the experiment 3.5, experiment 3.5, page 63 of textbook. So we're going to find out how to explain the methods to handle the variables, how to state the hypothesis, and then how to state the operational definition for a particular scientific term. So you're going to learn the secret techniques and even how to remember them. So stay tuned. Okay, let's continue. So you can be asked to explain the method of handling the variables, how to handle the variables, such as manipulated variable or MV, responding variable or RV, and fixed variable or FV, or can be sometimes called the constant variable, as in the Form 5 textbook, constant variable or CV. Okay, now in the exam, don't write MV, RVs, FV. You must write it in full if you are using these terms, manipulated variable, responding variable, fixed variable. So as we have discussed in the previous video, your MV is the concentration of sucrose solutions. Your RV is the final mass of the potato strip. This I refer to as sub-RV. My sub-RV here means that it is the RV that we obtain from the experiment. It is the data of the experiment, all right, the results of the experiment, the direct results. And then the main RV means the one that you obtain after calculation, for example. That is the percentage change in mass of the potato strip, the main or final RV. And that one we use when we write the hypothesis. Okay, put in the main RV, which is also stated sometimes stated in the objective of the experiment. Okay, that's the main RV. FV is the duration of time taken to immerse the potato strips in the sucrose solution. Okay, now pause the video for a while and try to write down, state how you will handle the different variables. Okay, so pause the video. All right, let's look at the answer. For MV, we can state use different concentrations of sucrose solutions, such as, let's say you know the values of the concentrations of sucrose solutions, then you can write such as 0.1 m, 0.3 m, 0.5 m, and so forth. Okay, so the keywords here are use different. Usually we use these words to explain how to handle the MV, but there are other types of format which we will not discuss here. This is the basic one. Okay, then for RV we use the words weigh and record, or this is weighing is for when you use an electronic weighing scale to weigh the mass, but if it is measuring the length of a substance or length of something, then you say measure and record. So it depends on what apparatus you are using. So here is weigh. You cannot say measure and record here. So weigh and record the final mass of the potato strip by using an electronic weighing scale. So weigh and record the RV by using the apparatus. Mention the apparatus when you are talking about RV. How you record the RV, the sub RV. Okay, so the keywords here are weigh and record or measure and record the RV by using apparatus. Okay, and finally for FV, we can write fix the duration of time taken to immerse the potato strips at a value. So mention the value, which here is 20 minutes. So we use the word fix the FV at a particular value. Try to find the value. All right. So for FV, the value is important. For RV, you must mention the apparatus. And here below, we see the apparatus used for the different variables. Here for MV, beakers and solutions, sucrose solutions. RV is the electronic weighing scale, and for CV or FV, we use a stopwatch. Let's look at the science process skill called hypothesizing. So you can be asked in this way: state the hypothesis for this experiment. 3 marks, right? Now let's learn the technique of answering. First of all, what are the components of a hypothesis? Okay, which you may have studied in the lower forms, right? So you must have MV, RV, and correct relationship. Okay, so in an experiment, sometimes you have 2 MVs, sometimes you have 1, and also sometimes you have 2 RVs, or you may have 1. It depends. Like in this case, the concentration of sucrose solution is our MV. There's only 1 MV, but we have 2 RVs, which I call the sub RV and the main RV, main or final RV. Okay, so these are my own terms. All right, sub RV for me means the RV that you obtain by doing the experiment. Is the data of the experiment, all right, that you obtain in the experiment. Okay, for example, our sub RV here is the final mass of the potato strip. All right, but you also have a main or final RV, which you calculated from the sub RV, and that is your percentage change in mass of the potato strip, like in this graph, right? Percentage change in mass of the potato strip. This is the graph that we discussed just now, right? So for hypothesis, it is better to use the main or final RV rather than the sub RV. Okay, because the hypothesis is related to the objective. At the beginning of your experiment, you must have an objective or aim, and you have an RV there. Usually is clearly stated. Sometimes it's not so clearly stated. However, your hypothesis will be related to your objective, right? And usually objective will use the main or final RV. Okay. Anyway, we try to use the main or final RV for hypothesis, which is the percentage change in mass of the potato strip, okay, which is calculated from the sub RV, okay, the final mass of the potato strip. Now next, of course, then your components for the hypothesis are MV, RV, and correct relationship. Now, the two formats for hypothesis. There are two common formats for hypothesis. The most common is this one. For factors that can increase or decrease, okay, the format is as MV increases or decreases, RV increases or decreases, depending on the experiment. Or the higher, the lower the MV, the higher or lower the RV. Okay. Now, the other format is for a few items that are compared, like fruits or food samples. All right. For example, determining the energy value of different food samples. So of all the MVs, a specific MV1 has the highest or the lowest RV compared to MV2 and MV3. Okay, for example, of all the food samples, groundnut has the highest energy value, which is your RV, compared to fish and dried fish and biscuit. Okay, we'll discuss more of this format later on. Okay, when we discuss the experiment involving vitamin C concentration. So let's go back to this one, format 1. We're going to use this to write our hypothesis here for this experiment. Okay, so pause the video a while, okay, and then recall back your MV-RV. Your MV is what? Concentration of sucrose solutions, right? Your RV is the percentage change in mass of the potato strips, right? And then find out the correct relationship. You can use this graph to guide you a bit. What's the relationship, right? Okay, pause the video and then write your hypothesis now. Right, let's look at the answer. As the concentration of the sucrose solution increases, the percentage change in mass of the potato strips decreases, right? Now, let's have a look. We can mark the components here. Concentration of the sucrose solution, one tick here, is your MV. Percentage change in mass of potato strips is your RV. Final RV. So one tick here. And the relationship is increases, decreases. All right. So one more tick. So 3 ticks. Now, why is the relationship increase, decrease? It's not increase, increase. It's increase, decrease. So it's like an inversely proportional relationship, but don't write the word inversely proportional here, or directly proportional, whatever. Inversely and directly proportional, we don't use it in biology. All right, we just write it in this format. Increase, decrease, etc. Don't use inversely or directly proportional. It may be used in other sciences, but not used in biology, right? So far. So here, let's look at the graph to understand our hypothesis. So here we can see the concentration of sucrose solution increasing from 0 until 0.5, right? Now, what solution is it that has a concentration of 0% sucrose, where the concentration of the sucrose solution is 0%? Actually, it's distilled water, right? Here, this one is distilled water, okay? Right. So let's look at the percentage change in mass. Now, as the concentration of the sucrose solution increases from 0 to 0.5, you can see that the graph goes downwards, right? So that means the percentage change in mass decreases from, say, 27% here for distilled water, positive 27%, drop down to 0%, right, for the isotonic solution, right? And then as we continue to increase the sucrose solution concentration, you can see that the percentage change in mass becomes negative and becomes a higher negative value, okay, which is considered lower in value, right? Like 20%, or somewhere here, 20%, negative 20% is lower than negative 10%. For maths, in maths, right? Because it's a negative value, all right? So our statement is true here. As the concentration of sucrose solution increases, the percentage change in mass decreases, right? Okay, now what if you were to write the sub-RV? It would be, so that's not so preferable. The preferable one is this, but if you would write it with the sub-RV instead of the main RV, it would be, as the concentration of the sucrose solution increases, the final mass of the potato strip decreases. Okay, also decrease. But anyway, keep to this one. Keep to this hypothesis using the main or final RV. Right, the last question here, how do we define a term operationally? Okay, this is another science process skill. So based on the results of this experiment, state the operational definition for osmosis. Okay, in this case, the term that we have to define operationally is osmosis, right? 3 marks. So this question on operational definition is usually more challenging for students. Students find some difficulty to answer this, but actually it's very easy if we know the technique of answering, right? So let's look at the components of an operational definition. First of all, the word operational means experimental. It's based on the experiment, all right? So the definition must be based on the experiment. But you first must have a basic definition from what you have studied, all right? From the theory, from the textbook, okay? The basic definition of osmosis must be there, must be used, and then you modify it to suit your practical, to suit the experiment, according to what is in the experiment. So if there's an organism, mention it, right? And if the term is a process, like osmosis is a process, so it's photosynthesis, respiration, or transpiration. These are all processes. Then mention the word osmosis is a process. Add it in. Then RV, you must add in the RV. So after you have stated your definition, write down, add on, and it is shown or determined by the RV. In this case, we are using sub-RV, not main RV, right? Sub-RV is the RV that you obtained, is the data that you obtained in the experiment, the raw data that you obtained through the experiment, okay? So that is the RV that you must use. Don't use the main RV or the final RV, such as the percentage change in mass, okay? So take note, for hypothesis, we use the main RV, but the main and final RV, which is the percentage change in mass. But for operational definition, because it's the experimental definition, you must use the sub-RV, which is the raw data that you obtain through the experiment, okay? And this is the final mass of the potato strip, the final mass of the potato strip. That's the sub-RV in this experiment. Now, CV can also be added into the definition, although it is not really counted yet as a part of the definition, all right, in the marking, but it may be in future, all right? So anyway, it's more complete to add in the CV, just a short phrase. And then MV, to add in the MV, we said RV is affected by the main MV. In this case, the MV is concentration of sucrose solutions. So you can write N is affected by the concentration of sucrose solutions, right? So in short, we can form an acronym, DORChem. Think of DORChem. D-O-R-C. The A is the word affected by, the words affected by, and M is for MV, DORChem. Like a door camera, a camera that you put near the door, right? But if for some teachers, maybe they don't include the C, all right? Okay, because it is not included as part of the points yet. Then you can, if you don't want to use the C, then put in the C, then you can shorten it to DOREM, like Doraemon, right now, DOREM. Okay, they'll help you to remember it more easily also. The DORChem or DOREM, up to you. If you want to put in the CV, it's DORChem. Without CV, it's DOREM. So pause the video for a while and try to write your operational definition for osmosis, okay, based on the components here. Right, let's look at the answer. Osmosis, Okay, start with the term. Don't start with something else. Start your sentence with the term that you are going to define. Osmosis. Osmosis is a process involving the net movement of water molecules in and out of potato strips. In and out of the potato strips, down the concentration gradient, okay, which is shown by the final mass of the potato strip after 20 minutes. And it is affected by the concentration of the sucrose solution, right? Full stop. So let's see the components. First of all, the definition, okay? Osmosis is a process. We mentioned that it's a process, and it involves the net movement of water molecules, right, in or out of the potato strips, because sometimes the water moved into the potato strips for certain concentrations of sucrose, okay, and sometimes it moves out, right, of the potato strips. So in or out, down the concentration gradient, okay, from region of higher water potential to region of low water potential, right, which is shown by the final mass of the potato strip. So that is your sub RV, very important. And the organism is mentioned here, potato strip, and also is mentioned in the sub RV itself. Okay, so after 20 minutes is your CV, and it's optional. And then it is affected by the concentration of sucrose solution. So your MV is the concentration of the sucrose solution, and the word affected by is here, right? So here the 6 components are complete already, are present. So you get the full marks, okay? Right. So I hope you have learned something from this video, some useful techniques to use in paper 3. Do share, like, and subscribe. And thanks for viewing this video. Until we meet again, goodbye.